Hello to all students. Today we are going to discuss transformation in bacteria. Transformation is a process in which naked DNA is transferred from one cell to another cell, usually bacteria. To prove this process of transformation in bacteria, Frederick Griffith, which is an English microbiologist, he performed some experiment on bacterium known as Diplococcus pneumoniae. He was a microbiologist and was working on to find the cure for pneumonia. Diplococcus pneumoniae is a bacterium which cause infection in animals and human beings and cause lung infection and blood infection, and due to these infection a host can die. Frederick Griffith used mice to demonstrate his experiments. First of all, he took a strain of bacterium which is known as S form of Diplococcus pneumoniae. There are some properties of this bacterium. Number one, this bacterium has polysaccharide coat on their outer body surface, which makes them very virulent, and due to this polysaccharide coat which is synthesized by their DNA, bacteria become resistant to immune system and immune system cannot kill them, so they cause infection in the animal and animal can die due to the infection by these bacteria. Number two, these bacteria usually form smooth colonies. When they combine, when they accumulate, when they multiply, they form smooth colonies. That is why we call them S form bacteria. Thirdly, they are very virulent. They can cause death, they can cause infection to their host animal. And because we are using mice in this experiment, so they can kill mice. So Frederick Griffith made a culture of bacteria in laboratory, put them in a syringe and injected into a mice. As predicted, mice got infection due to this bacteria and mice died. In another experiment, Frederick Griffith used another strain of Diplococcus pneumoniae which is known as R form. R form usually do not have polysaccharide coat on the surface of bacterium and there is no capsule. These bacteria form rough colonies when they multiply, and these bacteria are non-virulent because they do not cause infection and these are killed by immune system of the host, so they spare mice. Frederick Griffith made a culture of these bacteria, put it in the syringe and injected it to the mice, and as predicted, mice remains alive because these bacteria do not cause any kind of infection in mice. Griffith goes one step forward. He took S form bacteria, but before injecting in the mice, he heat killed these bacteria in a test tube. So heat killed S form bacteria are S form bacteria, but they cannot cause infection because they are dead. Griffith injected these bacteria into the mice, and as you know that heat can kill the bacteria and their virulency has been finished, these bacteria will remain alive. In another experiment, Griffith took now R form, which is non-virulent and usually does not cause infection in mice, and heat killed S form, which also does not cause infection in mice. He combined these two forms, R form which is alive and heat killed S form which are dead. In a previous experiment, both of these strains do not cause infection and mice remains alive. But in this experiment, what happened, when Griffith injected alive R form and heat killed S form together injected into the mice, mice became dead. So what happened? Because both of these strains are non-virulent and they do not cause the death of the host. But this time it happened that these strains together killed the mice. So what happened actually, which Griffith could not explain it, later on some experiment were performed and it was proven that it was DNA which was transferred from one bacterium to another bacterium. So what happened? A piece of DNA from dead S form heat killed bacteria transferred from this bacterium into R form which were alive. This DNA, this portion of DNA, when incorporated into the R form of bacteria, these bacteria developed a kind of polysaccharide coat on their surface and they become virulent. And this virulent bacteria, which become later S form, they killed the mice. So in this way, a transformation principle was confirmed. Transformation is a process in which a DNA, part of DNA is transferred, naked DNA is transferred from one cell to another cell. So I hope it makes sense and I'll see you in the next lecture. Until then, bye.